is my pleasure to introduce Dr. Helen Zhao. Is that correct? Zhao, or is it Zhao? Zhao?
Zhao.
Zhao.
Very
Good. I got it right.
Yes.
So she's an associate professor at the Center for Sleep and Cognition and the deputy director of the Center for Translational MR Research at the Yung Lu Lin School of Medicine in the National University of Singapore. Is that the new place or is that the old place? Did we get it right?
That's the new place.
Okay, good, good. And she's also affiliated with the Duke NUS Medical School. So she looks at brain network-based vulnerability in neuropsychiatric disorders. She does multimodal imaging, machine learning. So you'll have a real treat today. And she's going to be talking about early brain network changes in neurodegenerative and cerebrovascular disorders, looking longitudinally. So without further ado, Helen, I'll let you take it away. And thanks again for joining us.
Thank you, Vince. Good morning, everyone. I'm Helen. Thank you for the kind invitation and introduction. And it's my great honor to be here. And thank you all for joining us during this very tough period. So in the next 45 to 50 minutes, I'd like to share with you our work on human brain network breakdown in early neurodegenerative and cerebrovascular disorders from a longitudinal perspective. As you all know, given the aging society worldwide, the number of people with dementia will roughly double every 20 years. Alzheimer's disease is the most common type of dementia. So in 2010 and 2013, Jack and his colleagues proposed a hypothetical model of the major AD biomarkers as shown in this figure. It starts with the extracellular amyloid beta plaques, intracellular neurofibrillary tau tangles, followed by brain atrophy, even during the cognitive normal stage. And these pathophysiological abnormalities precede memory decline and clinical symptoms for many years to decades. Given this model and the failure of multiple drugs targeting late-stage dementia, the long-term goal is to predict the disease vulnerability and progression from a clinical and neuroimaging examination in every individual years before the appearance of any symptoms. To achieve this goal, as a first step, today I'm going to introduce network-based neurodegeneration hypothesis using network-sensitive imaging methods. Then under this framework, I will focus on three key questions. Can we detect symptom-specific changes in dementia subtypes? Can we use network-sensitive imaging methods to track the longitudinal changes in pre-dementia stages and the influence of risk factors? And lastly, can we review disease mechanism and predict disease progression? To start, here are the 5 T1-weighted structural MR images of patients with 5 different neurodegenerative syndromes. From the top, you can see Alzheimer's disease patient. In short, AD. So AD patients usually lose their episodic memory and visual spatial function, cause atrophy in the posterior parietal, posterior cingulate cortex, and hippocampal temporal network. Behavioral variant frontotemporal dementia, in short BVFTD, in contrast, disrupt the social emotional functions and target an anterior frontal insular and anterior cingulate cortex and subcortical network. As you can see, all 5 disorders have very distinct atrophy patterns. Based on the evidence from neuropathology, imaging, and animal models, neurodegenerative diseases have been thought to relate to a network-based dysfunction. In other words, each disorder targets a specific large-scale neural network. In 2008, Bill Seeley led the team to systematically test this network-based degeneration hypothesis in living humans for the first time. We used a network-sensitive neuroimaging method, which includes resting state fMRI and structural covariance network. I guess this group does not need the introduction of resting state fMRI, but to be very brief, task-free or resting fMRI captures the blood oxygenation level dependent signal fluctuations at rest. We can derive the intrinsic connective networks, so in short ICNs, from these four-dimensional data, which map the temporal synchronized, spatially distributed, spontaneous low-frequency signal fluctuations. We can derive both higher-level cognitive networks, such as default, control, or attention networks, and low-level sensory motor networks. So, starting with the atrophy pattern of each of the 5 disorders, as you can see here, highlighted in cyan color, we pick the most atrophied regions circled here, for example, right frontal insular for BVFTD or right angular gyrus for Alzheimer's disease. By doing the seed-based functional connectivity mapping in healthy participants, here are the resulting ICNs, as highlighted in the yellow color here. You can see great similarity between functional connectivity networks in healthy controls in yellow and atrophy pattern in disease. Similarly, if we use the same set of seeds using structural covariance network based on structure MRI in healthy controls, you can see similar pattern highlighted in green color. There is a direct link between the functional networks and gray matter covariance structure in healthy participants. And taken together, we have confirmed the network-based neurodegeneration hypothesis in living humans. In other words, regions wired within each network, they fire together, they grow and shrink together. When the disease starts, these regions also start to have atrophy together. This work leads to many interesting questions on selective network vulnerability along disease progression. Following Jack's model, Beers and other colleagues proposed and refined this model by including functional connectivity as well as FDG-PET metabolism. As you can see here, these two, together with many other biomarkers, have very differential trajectories from preclinical to dementia stage. So now let me start with the first question. Can we use these techniques to detect symptom-specific network changes in dementia subtypes? AD targets the deformer network specifically. This is supported by increased amino burden from PET, atrophy from structural MRI, and hypometabolism from FDG-PET. In 2004, the pioneering work by McVicious and colleagues from Stanford found reduced deformer network connectivity in Alzheimer's disease patients compared to age-matched control using task-free fMRI. And note that this is independent of brain atrophy. How about the other dementia subtype? Indeed, our previous work have shown that AD and BVFTD have divergent functional connectivity changes in the default mode network and salience network. Specifically, BVFTD patients have reduction in the salience network, but increased connectivity in the deformer network, while AD shows the opposite pattern. These divergent patterns correspond very well with the symptom deficit profiles of AD and FTD, which again supports the network-based dysfunction hypothesis. Since then, our findings have been replicated in quite a number of studies, including MCI or APOE4 carriers. One step further, we hypothesized that the network coordination between these two networks, as well as subcortical regions, is differentially disrupted in AD and BVFTD, which may lead to cognitive and social emotional deficits. We test this hypothesis in a single point cohort of AD and FTD. Based on the whole brain pulsation, we found that the community structure reconfigurations has changed in both AD and BVRPD compared to age-matched healthy controls. When we narrow down to specific functional modules, as you can see here, AD have more fragmented modular structure in the default and executive control network, while FTD had more severe damage to the modular structure of the salience and subcortical structures. Importantly, such connectivity profile track the disease severity and clinical symptoms. Across the FTD patients, lower salience network functional connectivity in right frontal insula in the y-axis here was associated with more severe disease severity measured by clinical dementia rating scores. And if we look at the connectivity between salience network and other networks, we found that the increased salience network participation coefficient, indicating this loss of modular structure or functional distinctiveness, was related to more severe behavioral problems as measured by neuropsychiatric inventory score. Our findings support the distinct loss of network integrity in these two disorders, underlying their opposite cognitive and behavioral symptoms. When we focus on Alzheimer's disease alone, recently, several works have tried to explain the clinical variability in AD. However, they only examine AD patients without comorbid conditions. But in fact, in reality, AD frequently coexists with cerebrovascular diseases, in short, CVD. AD with CVD is the most prevalent dementia subtype. On its own, CVD is associated with decline in executive function and processing speed. CVD MR markers include lacunes, 1-minute hyperintensity, and microbleeds, etc. Previous studies have demonstrated that CVD and AD could have these additive effects on cognitive decline. From a pathology study, if they have the same clinical severity of dementia, AD-only participants will have more severe AD pathology like amyloid and tau than those comorbid with CVD. So Joanna Chong from my lab asked this question: can we see a similar or differential functional network breakdown patterns in AD patients with and without cerebrovascular disorders? Based on the memory clinical cohort from Singapore, for the first time in the field, we showed that AD patients with and without cerebrovascular disease featured divergent functional connectivity changes in the default and executive control networks. When we compare the CVD and non-CVD groups with the same disease severity head-to-head, AD had more reduction in the deformal network connectivity, which related to lower hippocampal volume. Similarly, CVD groups have higher frontal executive control connectivity, which was correlated with greater burden of CVD as measured by 1-minute hyperintensity. This work supports the differential network degeneration phenotype in AD with and without CVD, which reflect the underlying pathology. Besides the functional network breakdown, white matter degeneration is also an early event in AD, which closely follows A-beta deposition. Diffusion MRI measures the movement of water molecules within the white matter tracts and can be used to examine the structural connectivity and white matter microstructure. So now I'm going to show some evidence of white matter abnormalities in AD and CVD. The traditional single tensor diffusion model suffers from a partial volume effect. Basically, in a one voxel, the signal there is a mixture of tissue compartment and water content. How do we solve this problem? We collaborated with Dr. Postulak from Harvard Medical School, who proposed a biotensor free water method. This method separates the signal of each voxel into two compartments. The tissue compartment represents axon bundles, and the free water compartment represents the extracellular water. The reduced FAT, or changes in tissue compartment, are related to axon damage and demyelination. Increase in free water might be caused by vascular damage or inflammation, possibly due to the blood-brain barrier leakage. So Jifang in my lab, in collaboration with the memory clinic, is interested in the differential white matter changes in AD with and without cerebrovascular disease. Compared to the original single tensor model, we can see that the free water correct tissue compartment revealed more accurate and focal changes in the frontal and occipital lobes, as highlighted in red color here. When we look at the free water increases across the three dementia subtypes, AD, AD with CVD, and vascular dementia, it is not surprising to see that the vascular dementia groups, as well as patients with cerebrovascular disease, has widespread free water increases compared to AD without cerebrovascular disease. But interestingly, if you look at the left panel, AD without cerebrovascular disease have comparable CVD burden compared to age-matched controls. But they have higher free water in the normal appearing white matter compared to controls, which indicate that AD without cerebrovascular disease still have neuroinflammation or microvascular damage. Importantly, such free water increases in normal appearing white matter, but not white matter high intensity, were associated with symptom severity across all dementia patients. Building upon this previous study, we continue to investigate whether such brain free water increases were related to cardiovascular markers and whether it could mediate the relationship between cardio burden and further future cognitive decline. Based on our longitudinal data from Singapore, what we found is that the higher cardiovascular markers, such as high sensitivity cardiotroponin T, was indeed related to free water increases in widespread white matter and network-specific green matter regions in the default control, attention, and sensory motor networks. When we look at the cognitive decline up to 5 years, we found that the white matter free water increases and these network-specific green matter free water increases modulate the mediated associations between the cardiovascular markers and the longitudinal cognitive decline, especially executive function and verbal memory. You can also see this nice dissociation where the executive control network influenced the executive function decline and the deformal network influenced the verbal memory decline. Such results underscore the important role of free water imaging and how the vascular modulation is related to the brain mechanisms. So taken together, I hope we can convince you that based on the structure, diffusion, and functional MRI, these findings actually lend further support to network-based dysfunction hypothesis at dementia stage. Brain network phenotype explain clinical variability, track disease severity, and relate to AD and vascular burden. Till now, I have reported mostly cross-sectional work in dementia stage. As you know, to facilitate early detection and early intervention, it is crucial for us to study how these network changes lead to early cognitive decline over time and the influence of risk factors such as ApoE or pathology. Again, based on our previous work published in Brain, we're very interested to find out the changes and in the prodromal stage, right? So mild cognitive impairment, in short MCI, is often considered a prodromal stage of dementia. In collaboration with some medical center, we're interested in whether amyloid and cerebrovascular burden are associated with differential network changes over time in MCI. And this time, we have a direct objective measure of AD pathology using amyloid PET imaging. Again, we focus on resting state functional connectivity over time in two networks. One is the default, the other one is the executive control network. To test the hypothesis, and Joanna Chong actually compared the two pure subtypes first. The so-called amnestic MCI A-beta positive is a pure AD subtype. You can see that every line here represents one participant, and the prodromal patients with pure amyloid burden showed steeper declines in the deformal network connectivity. When we look at the subcortical vascular MCI, A-beta negative, which is usually considered a pure prodromal stage of cerebrovascular dementia, this participant, as highlighted in the salient color here, showed steeper increases over time in the executive control functional connectivity. Then you can see that if actually we include all other participants with mixed pathology, such as amnestic MCI with amyloid negative or subcortical vascular MCI A-beta positive, and repeat the whole analysis in a continuous manner, we found the same thing. Basically, amyloid burden had an effect on the longitudinal default mode network functional connectivity changes, while the Lacoon number had an effect on executive control network changes over time. Now let me move to the very early preclinical stage here. The key question is whether the longitudinal network changes could reflect the cognitive decline even at the early stage. Let me take 1 minute to talk about the modular organization of the brain. We know that the human brain is functionally organized in a highly modular and distinct manner, with high connectivity within network, indicating functional specialization, and low connectivity between networks, indicating functional segregation. Such modular structure can aid the specialized and integrated information processing, increase the robustness and adaptability of our brain networks. For example, the task-positive networks, the salience network, and executive control networks are often anti-correlated or segregated from the task-negative or deformal network. The salience network is also closely connected to the subcortical regions and executive control networks. So given the multidimensional nature of cognitive decline, we hypothesize that both within and between network connectivity will be important for cognitive deficits or decline. So to cover this gap, actually my postdoc fellows Eric Ang and Qiu Yingwei studied the longitudinal changes in functional connectivity and cognition over 4 years in a group of Singaporean healthy older adults aged 50 to 85. What we found is very interesting. If we look at the intra-network specialization, we found that across all 7 networks, we actually see specific functional connectivity decline within the default mode and executive control network over 4 years. When we look at the functional connectivity between the networks, interestingly, aging is associated with this age-dependent decline in the segregation between the default and executive control network. So each line represents one participant. You can see that after age 70, we actually see this loss of functional segregation over time. When we divide the participants into APOE4 carriers and non-carriers, you can see carriers are highlighted in red color here. APOE4 carriers who are at a higher risk of developing dementia had faster loss of functional segregation over time compared to non-carriers, suggesting that they actually have this accelerated aging pattern. If we extract the annual rate of changes from processing speed and annual rate of changes in the functional connectivity between executive control and deformal network, we found this nice association indicating if you have greater loss of functional segregation between these two networks, you are going to have faster decline in processing speed over time. So in a very recent study published in our lab, we actually consider the whole brain functional network organization. So Joanna Chong, using this global index of network distinctiveness, we found that elderly show this cross-sectional and longitudinal decline in network segregation and distinctiveness. And when we do multiple comparison correction across all 9 networks we're looking at, we can see that such changes are very specific to the higher order cognitive networks, including the salience, executive control, and deformal networks. So now let me change gear again to talk about early white-matter degradation in pre-dementia stages. Again, we're interested in the influence of cerebrovascular disease and amyloid burden. Based on the ADN database, Ashwati Vipin from my lab used the free water imaging and derived the free water as well as the free water corrected tissue compartment using diffusion MRI. When we look at the cognitive normal elderly and separate them into amyloid positive and amyloid negative, what we found is that amyloid positive individuals, highlighted in the red color here, have a faster reduction in the fractional anisotropy in the right ancillary fasciculus over time than those amyloid negative participants, which is suggesting that there is this amyloid-related early white-matter degeneration, especially on the fiber tracts linking the frontal and temporal regions. In terms of CVD burden, in collaboration with our Leiden colleagues, we're interested in this novel cerebrovascular the marker, the so-called cortical cerebral microinfarcts, or CMI. So CMI is more prevalent in AD and related to cognitive deficits. Animal models suggest that the CMI induced white-matter impairments on neuronal loss and decreased neuronal responses in remote cortical areas beyond the actual region of the cortical CMI. Therefore, we hypothesize that the cortical cerebral microinfarcts may affect the cognition via disrupting the local and long-range white-matter pathways. So this is the first study examining this question in living humans. What we found is that the presence of cortical CMIs led to lower structural network efficiency and degree centrality in the executive control and dorsal attention networks. This is true in a pre-dementia stage, but such effect was missing in the dementia stage. And one step further, we found that the structural network breakdown mediates the association between cortical CMIs and cognitive decline, which support our hypothesis that the CMI actually influence the cognitive decline via the structural network breakdown involving remote regions, especially at the early stage. So lastly, we're going to answer the last question. Can we build up disease progression predictive model based on multimodal brain measures? One specific question we have asked before is that whether such brain network measures can give us insight into the disease mechanism and progression. We have demonstrated this possibility at the group level previously by comparing a healthy connectivity graph theoretical measures to atrophy severity in disease. So basically, if we look at AD, we have demonstrated that regions having shorter functional paths to the disease epicenters in health would have greater disease atrophy when the disease starts to hit them. So when we look at these measures across all 5 different dementia subtypes, we are able to replicate these findings. The first row indicates the same finding. Shorter you are to the disease epicenter, you are going to have higher atrophy in disease. The second row represents the total flow. So the more traffic you have through a certain region, you are going to have higher atrophy in disease. So these two support the pre-online transneural spread model or the nodal stress model, but did not support the trophic support model. So our findings are consistent with several previous studies in the sense of basically the amyloid deposition is associated with degree centrality, which support the nodal stress model. Another finding using diffusion imaging and do the simulation, which demonstrate that the closer you are to the disease outbreak region or disease epicenters, actually modulates the amyloid propagation process. So the closer you are, the earlier arrival time of this toxic agent, such as A-beta. However, all the previous works are based on group level studies. Recently, we would like to actually look at whether this particular framework can be extended to the individual level. So this work from Jesse Brown, they actually show that rather than the group level disease epicenter, there is this unique atrophy epicenter in individual level, which is vary across participants, right? And they then use such patient-tailored epicenters in a connectivity-based model to accurately predict the individual longitudinal spread of atrophy. So these findings support the transneural spread model at the individual level. Under this framework, our lab also have made some effort to derive individual level network integrity score. And Qianxin from my lab is particularly interested in whether and how the influence of network-based neurodegeneration on cognitive function vary with different disease stages, as hypothesized by that AD hypothetical model. So to answer this question, we derive the individual level metabolic network score from FDG-PET, as shown on the top panel, and structural covariance network score from structural MRI using partial least square. As you can see, both networks nicely resemble the key canonical brain networks derived from resting state fMRI. Using some statistical modeling, we demonstrate that there is this stage-dependent association between network integrity and cognitive decline in AD continuum, from cognitive control to MCI to probable AD. And such trajectories are differentially affected by pathology. And another example of the network breakdown model is in stroke patients. We follow the stroke patients from 3 months to 1 year post-stroke, and using structural MRI data, we're able to demonstrate that if patients have faster degradation of brain structure networks, they are going to have a greater longitudinal decline in attention, language, and memory. Here again suggests the potential of using brain network integrity to predict and monitor disease progression. The last example, in the spirit of predicting disease progression, we know that only a small proportion of individuals with at-risk mental state for psychosis eventually progress to clinical psychosis. So it's critical to predict the conversion in these individuals to facilitate early intervention. So based on a numeric study from Singapore, we found that the baseline functional and structural disconnectivity in the salience network can actually predict their future conversion. You can see a huge differences between ARMS participants who transition to psychosis 3 years later compared to those who don't. And note that this is data 3 years before at the baseline. So this observation of unifocal connection-based spread is only one of the possible network breakdown scenarios. It does not take into account the genetic vulnerability and other biological factors. So it can be very well be a staggered multifocal onset, which is not related to functional connector. Or it could be a combination of the two. So we really have to integrate neuroimaging, genetic, and pathological data from both animal and human models to further test this network-based breakdown hypothesis and its behavior and its relationship with behavior and symptoms. So in conclusion, I hope I have convinced you that it is important to examine early and longitudinal brain network breakdown trajectories in neurodegenerative and cerebrovascular disease. Moving forward, longitudinal multimodal neuroimaging methods, together with genetic, blood, and CSF, or digital tracking with high reliability and reproducibility, will allow us to formally test the predictions made by network breakdown hypothesis. We'll also need machine learning and other computational approaches to discover robust and valid features with high accuracy, generalizability, and interpretability in order for us to improve subject stratifications and facilitate prognosis and early intervention. And just one example is we all know the problems of resting state. The vigilance do fluctuate over time. So using dynamic functional cognitive models, we are able to demonstrate that there is this time-varying recurring cognitive patterns related to varying consciousness levels, vigilance levels, and individual differences in the behavior, which is also across individual differences outside the scanner, as well as modulate their changes temporally. So with that, I would like to thank the funding agency and all the collaborators and our lab members who have contributed to these studies. And thank you for your attention. I'm happy to take any questions.
Thanks, Helen. That was great. So do folks have questions? You can use the chat or the Q&A section, or you can also let us know and we can unmute you and have you ask your own question if you'd like. One that came up was, this is from Armin, which is how accurate are these multimodal features? How accurately can these multimodal features in network-based models classify Alzheimer's disease in the early stage?
Okay, so this is a very good question. So I guess you are asking about the machine learning, how accurate they are, right? So nowadays, I think I have seen quite a number of studies using structural MRI. They are able to achieve around 80. So if you're asking about a classification between 80 and control, they can achieve an accuracy of 80. Then if you're using the PET imaging, like those with amyloid and FDG PET, you can even achieve higher than 90%. But if you're asking about a specific question, let's say discriminate MCI who are going to convert into AD and those who are not going to convert into AD, the accuracy will be lower, around like 70 to 80. So it really depends on the question. That if you go to the very early stage, like the preclinical stage, and predict who is going to convert later on, the accuracy is even lower. So there is this famous Tapo challenge. Rather than predict basically who is going to convert, they basically predict the amount of cognitive decline or clinical dementia rating score. So they basically evaluate the MSE or the R values. So I believe there is still room for improvement. You can check this out.
One just comment to that is there's a paper I think it was written by Anis Abrul from our group, using deep learning, and showed on gray matter maps that I think predicting AD from controls was over 90% with just structural MRI, and conversion from MCI was in the, I think, around 70 to 80% range. So yeah. Jess Turner says, marvelous work. Thank you so much for those interesting ideas. So, how much have you looked at? I know you were looking at the structural covariation, and have you done that jointly in PET? And is that what you were doing with the PLS? Was PET and gray matter?
Yes. Yes.
Yeah.
I haven't done it jointly. I have done it separately.
Okay.
But I know you have this very fascinating multimodal joint ICA model, which can extract the covariance from the two modality together. Yeah, we would like to try that as well.
Yeah.
Yeah. Yeah.
Yeah, it's cool. It's very interesting, and I think those links between gray matter, maps and functional measures too are fascinating, I think. And
yes.
Yeah. Other questions? So how close are we to having this be something that can be used? I'll just ask the elephant in the room. So
yeah. So I think that I think actually the amyloid PET imaging and RPT PET imaging has already been included in the diagnosis of the Alzheimer's disease. Right. So right now the ATN hypothesis is you have to actually look at the pathology, then you are able to determine their trajectory or predict their disease progression. If you're asking whether we can use imaging to help us to do the prediction, and the accuracy, I think we still need to do a bit more. For example, I think right now we already know this quite distinct network patterns in different dementia subtypes, for example, and then we need actually some generalizable machine learning model to able to capture that. Because previously, even though I showed this distinction, but when you apply to a new participants, the model usually cannot predict very well because the atrophy and network changes vary across the participants. And where the disease starts also vary across the participants. So I think the individual level patient-tailored model will be very important. And that's why I said longitudinal data is very important. We have to have longitudinal data. And secondly, we have to have the data very early. And we have to have multimodal data. And then obviously the genetic data is very important. So yeah, then the machine learning models. If we can learn automatically from the data itself, it will be much better. And then lastly, the interpretability, right? Because if we do it and we don't know what is going to help us to do the discrimination better, then it may not be very attractive to clinicians.
What's more important, more data on one subject or more data on many subjects? Less data on lots of subjects, or very dense data on fewer subjects?
That's very
come up in some recent work, right? So yeah.
That's a very, very nice question. Both are important. I mean, deep phenotyping of a small number of participants, but many, many data points obviously can help us to discover a lot of things. right? So we would like to do that as well. However, in terms of generalizability, then and machine learning, from machine learning point of view, then you need a lot of data for sure. Yeah. And you definitely have to look at the reproducibility and replicating your results across different cohorts. Based on my experience, the nature of the brain network changes across different ethnicity or different gender or different genetic vulnerability could be very different.
Okay. Great. Well, thank you. So just as a reminder, for the BrainSpace Initiative, we have this Slack channel, and the video will be was recorded and will be released. And the Slack channel will be for this talk, and so there can be an opportunity to kind of interact if folks have follow-up questions and they want to, get a little bit deeper into it. Oh, we've got one more question here, which is from Brian Sadler, which says, analogous to a communications network, does the connectivity data reveal a communication rate decay? Do we neurologically adapt to the reduced networking by, say, changing the message? So that's more getting at function, I guess, right?
So the question is regarding network communications from fMRI. Am I able to see the?
No, I think it's not specified, but I would think anything functional, maybe. Yeah, could be fMRI, could be something else.
Okay.
I think that's a great research question, and I look forward to reading your paper, Brian.
Okay, can you probably repeat the question, because I didn't catch what is
sure. So analogous to a communications network, does the connectivity data, so I guess it is talking about like functional connectivity, reveal a communication rate decay? So is the communication changing in its rate, for example?
Yes.
And then he follows it up by saying, do we neurologically adapt to the reduced networking by changing the message? So does the network atrophy and does the communication change?
Okay. Yeah, I guess I didn't get the first communication network thing, and then that's it. Okay, I know what you're talking about, and I do think there is certain kind of adaptation here. We do see a lot of U-shaped changes, if that's what you're looking for. So that's why I actually moved from pre-dementia to the preclinical stage, because some of the functional connectivity patterns we see, when we move to the very early stage, you don't see that, and you see this U shape. They're going up and then go down, right? So the network segregation is one example. You see this U shape, so-called age-dependent loss of functional segregation. Before 70, you actually see this increased segregation between the network, suggesting that because they have this loss of functional specialization within the two networks, therefore they're trying very hard to maintain the segregation. And then after 70 years old, nothing works. Everything breaks down, right? Loss of functional specialization, also loss of functional segregation, so it goes up. So I do believe there is certain kind of adaptation in terms of how they communicate across the networks. And then once, let's say people have reached their limit, then this network starts to change significantly.
Thank you.
Yeah, just to add, the free water methods also illustrate that. Even though this is microstructure changes, but we also see very, very early stage, the free water goes down and then goes up.
Brian, you can follow up if you want. You're unmuted.
Oh, I appreciate that. I'm not a neuroscientist. I'm more of a intelligent systems and multi-agent networking guy. So I was kind of curious. I realized the kind of sensing you're doing is limited in terms of things like, what I would make an analogy in communications network with, say, rate, right? So we might have multiple agents that the rate starts to break down. So we might have to adapt the message or go to a different level of abstraction. That's kind of what I was poking at.
Yeah.
Yeah. And I do appreciate the answer from my naive perspective. Thank you.
Yeah, I think it's actually a very good way to think about things. I mean, we've certainly looked at things like entropy and also frequency, which changes. We've looked at it, for example, in mental illness versus controls, and it's clearly different, and you see different relationships and different amounts of information, right? So I think modeling it within a communications framework would be certainly quite applicable and interesting. So. Yeah.
Yes, I think some people use atrophy and look at the EEG data and see a very diverse pattern in FTD and AD. So I think you definitely should have a try.
Yeah.
Thank you.
Great. Okay, well, thank you, Helen. And we will follow up, as I mentioned, with the Slack channel and let everybody know. And Armin, anything else?
No, thank you so much, Helen. I really appreciate that.
Yeah, we'll let
Thank you so much.
We'll let you go to sleep or not. So
okay, thank you so much, Vince and Armin, and I very enjoy the discussion. Yeah. Okay, I will follow up. Yeah. Thank you.
Bye-bye.
Good day. Bye-bye.